Hello, and welcome to the Talking Precision Medicine podcast. In this series, we sit down with experts on the application of AI and big data analytics in the drug discovery space. Our guests are innovators, business decision makers, and thought leaders at the intersection of data and therapeutics. We discuss the promise, practice, challenges, and myths of AI in precision medicine. This show is brought to you by Genialis, and Rafael, our CEO, is your host. Genialis is focused on data integration and predictive modeling of disease biology to help accelerate the discovery and de-risk development of novel therapeutics. Today we speak with Olivier Elemento. Oliver is the director of the Englander Institute for Precision Medicine, as well as associate director of the Institute for Computational Biomedicine, both at Weill Cornell Medicine. He is also co-founder of OneThree Biotech. Oliver is a practitioner par excellence in applying advanced analytics to large genomic and patient datasets. Our conversation covered a lot of ground, including curated knowledge bases, patient consent, breakthroughs in preclinical models, and even AR/VR. Let's get right into it. So I'm here today with Dr. Olivier Elemento. Olivier is the director of the Carroll and Israel Englander Institute for Precision Medicine. He's associate director of the Institute for Computational Medicine. These are both at Weill Cornell Medicine in New York. And he's a co-founder of a new and exciting startup, OneThree Biotech. So Olivier, that's a ton of stuff going on. Let's start at the top. Tell us more about the various roles you play in running your lab, directing the institute, starting a company.
Yeah, so as you mentioned, I do a few things here at Cornell. So what sort of occupies most of my time is running the Institute for Precision Medicine. So that's an institute that has two missions. One is a clinical mission, which is to use cutting-edge genomic analysis to basically, identify how we can help patients using genomics, how we can sequence the DNA of, tumors, the germline DNA of patients, for non-cancer patients, and use that information in a way to optimize how we treat patients, how we care for patients, how we diagnose disease, and so on. And so this is something we've been doing for several years now. We have some really cutting edge assays, in that space. We're running whole exome sequencing of tumors, sort of tumor normal sequencing 22,000 genes for every tumor that we sequence. And it gives us a very detailed, view of the mutational profile of tumors. And that gives us a lot of options, gives our physicians a lot of options in terms of what treatment to use. We're focusing on advanced cancer patients, basically patients whose tumors evolved to the point where it's, resistant to chemotherapy. It's often, metastatic disease, multiple locations. Surgery is not an option anymore. Radiation is not an option. So we're looking for targeted therapy options for those patients. We're looking for, NTRK fusions, which have recently become actionable, MSI high, high tumor mutation burden type of situations. And looking also to use, sort of basically off-label, sort of drugs as much as possible. If we can find mutations that we know are important in other tumors, maybe not, with an FDA-approved drug in that particular tumor type, but there's basically a drug that's approved in a different tumor type where those mutations are found, then potentially we can give those drugs to patients, really, so last resort type of options. We put patients on clinical trials, quite a bit as well, as a result of this genomic analysis. So, the primary goal is a very clinical goal, and that's, it's working really well. I mean, the strength of the program is that as we generate a lot of information about patients, as we sequence, thousands and thousands of cases now, we are actually starting to get a lot of information. We have an extensive database of genomic profiles, and these are patients treated here at Cornell. So, they've consented for us to also use not only the genomic, data for research, but also the clinical data. And so we're starting to make very compelling, interesting, novel associations between genotypes and phenotypes. And that, is giving rise to some interesting associations. And in some cases, it's starting to give rise to potential, drug targets. We have industry alliances also as well that allow us to capitalize on these discoveries, and basically sort of get to the next step, which is to try to come up with drug candidates, basically based on the targets that come out of the effort.
That makes a lot of sense. Can you talk a little bit more about the clinical side? So Cornell has its own clinical facilities there. Are you working with some of the bigger New York hospitals? How does that work?
So we are part of a sort of hospital, sort of system here. It's called New York Presbyterian Hospital. It's, one of the largest hospitals in the US. So we're basically a medical school that's connected to a hospital, and we work together very closely with the hospital and many things. So we run our own clinical lab. So we have a CLIA lab that, where we run the assays, that we develop. We have to go through New York State approval for all these assays. What that means essentially is that we have to credential, these genomic assays, basically, so demonstrate that these assays, produce reliable information, reproducible information. We have to report the results, back into the EHR, which is something that's also very unique to what we do. We have a full integration between genomic analysis and the electronic health record. Physicians order testing in Epic, which is the clinical system that we use, and the results of the genomic analysis go back into Epic. So that's where they consume the information. That's where essentially they can find the genomic reports. But in addition to this, the strength about using the EHR is that we can essentially connect in a very sort of easy way clinical data and genomic data because it's already in the EHR. And so we use I2B2 as an example as a way to do queries that combine genomic information, genomic mutations, and clinical data, demographics, and diagnosis, lab results, and so on. So the paradigm is very powerful, and we are actually one of a few institutions in the US actually that have enabled this integration, this very deep integration between genomic analysis and the EHR. Very proud of it, and it's really the way to go. It enables our physicians to consume the information in much the same way that they consume every other type of results that they get from their patients, basically through the EHR.
And so who's actually performing the secondary and then tertiary genomics analysis? Is that happening in your lab or in a group of labs? How is the bioinformatics and then, of course, the more advanced analytics done?
Yeah, so we have it's an entire workflows. It's a set of tools that we use. We've actually at this stage automated the whole process, pretty much the whole process of analysis of results, interpretation of mutations. We're using a resource that we created here at Cornell. It's called the Precision Medicine Knowledge Base. It's a database of cancer mutations, but focusing on actionable cancer mutations. Basically, mutations that are important when it comes to treatment, when it comes to diagnosis or prognosis, and so on. So we maintain our own database, which is, very sort of focused on the kind of mutations, the patient profiles that we see here. But it is also made available to the public. This is a resource that we're trying to make available to everyone on the planet, basically, and it's used actually across the board, and just by many people across the planet. And so that actually is very helpful. Essentially, more or less at this stage, is still, sort of looking up, sort of information. So every time we sequence the genome, we get a list of mutations, point mutations in there, copy number alterations, translocations if we do RNA-seq, and we look up all these mutations in our database. And when we see a match, we basically have an interpretation, a clinical grade interpretation that's connected to the mutation that's also tumor type specific, and that we report on the report. Our pathologists, the clinicians who sign our cases do make maybe sometimes minor modifications to the report of interpretation, but we basically generate a pre-report, if you want. That is almost ready to be signed out, but can be modified, if pathologists want to modify the reports. So we're trying to make it as easy as possible to sign our cases, but also really to put the results in a format that's easy to digest by clinicians.
So you're making this available to the public. This is the Precision Medicine Knowledge Base at Cornell. If a lab from the public or another organization were generating a lot of this data, would they have a way of submitting to this as well, or is there a way that you can kind of trust what they've done and curate that?
Yeah, so we're trying to start this system where we would allow submissions from outside. Obviously it's a clinical database, so we have to vet the information very carefully. So we have a committee of pathologists here who essentially vet the information that goes into the database. So there's like an approval process, if you want. It's we want to make it easy for people to submit information, but also we want to make sure that only the very sort of well vetted information goes into a final database. So we are basically working on this kind of system where we would allow submissions from the outside, still enabling vetting from a pathologist or from maybe other pathologists. I think we feel that the information that goes into the database at the end of the day has to be vetted by board certified pathologists, just because, they know, what information, should be there, and they know how to, sort of, describe it, they know how to, what language to use in those interpretations. And we feel that they really should have a last word, if you want, in terms of what goes into the database.
You mentioned just kind of in passing that you have a New York State regulatory burden. How is that similar or different from, say, a federal regulatory burden?
Yeah, so it turns out, the basically getting approval for genomic testing is very different, between states in the US. In New York, we actually have the highest level of sort of burden, if you want, in terms of getting approval for genomic testing. We have to provide a gigantic amount of information about, the test, where it's performed, who performs it, training, procedures, material, and so on. But also, essentially, we have to provide a lot of information about the test itself, the limits of detection, the different types of tissues that the test can be used on. We basically have to demonstrate that the test is reliable in all kinds of different conditions, whether it's, let's say, low purity tumors, but paraffin embedded sort of tissue as an example. We have to demonstrate that we can do genomic testing and get reliable results in all of these different conditions. And so that would complicates very much obviously the sort of the submission process, the certification process. But you know what? I mean, it's actually a good thing. It's actually good for patients that they get access to a test that's vetted to this level. We think it's even though it's a burden, it's actually extremely good for the patients. And so we are quite sort of happy about the process in some ways. I mean, the process is actually not really fundamentally too different from what it takes to get to try to get FDA approval. So it's actually it's in New York State, it's very difficult to get approval for genomic testing. We see it as an achievement that we've been able to get approval, for example, for whole exome sequencing. We are actually the first institution to have gotten approval for whole exome sequencing, tumor normal in the US.
You've talked a lot about whole exome and the sort of genomic information. What other kinds of data, either omics or otherwise, so you've got, I guess, the clinical data as well, the EHRs, but what additional information are you collecting on these patients, or maybe does your lab think about working with in addition to these data?
Yeah, no, so that's a great point. We really think of the future of genomic testing and analysis of tumors as, obviously multi-omics. So, I mean, right now, for a variety of different reasons, it's easier in some ways to do systematic analysis of the DNA. The DNA is very stable. We have some really good assays, to interrogate the DNA. But really moving forward, we really feel like we need to have also, some kind of functional, let's say, readout of a genome. We're trying to do RNA-seq, basically transcriptomic analysis of tumors as often as possible. We started this using essentially only frozen tissue. That's where it used to be, sort of most practical to do RNA-seq. But in the past, couple of years, we started optimizing RNA-seq from FFPE tissue, which allows us to essentially now almost systematically do RNA-seq in our patient samples, which I think is going to be a game changer. We've actually published several papers really showing that RNA-seq in combination with DNA sequencing, especially whole exome sequencing, gives you a much more compelling, much more sort of comprehensive readout of a genome, and really sort of, adds a lot of actionable, targets, actionable mutations, to the genome. For example, it gives you a sense whether a mutation is actually, a driver mutation or not in many cases. If you see a mutation in a gene that's not expressed, very often it's basically not a driver mutation, just as a result of basically the gene not being there. If you see that, the genes that are known to be downstream of a mutation are not expressed, it basically means that, maybe the mutation is there, but it's not doing anything because the genes that are known to be downstream are just not doing anything, basically flat. So, I think there's, we're just starting to get into this. But in my view, I think there's a great future towards combining DNA sequencing with essentially a functional readout of a genome. And I think RNA-seq is, one of those sort of assays that I think will be very useful and practical when it comes to getting that functional readout. It really sort of gives you additional, let's say, confidence about, the fact that the mutation is really doing something that you expect it's doing. So that's, RNA-seq is one of the things I would like to be able to do in all tumors. We don't have a clear assay yet, for RNA-seq, but I think that's something we'll prioritize in the next sort of few months or next few years, because it's really important. But we are still now doing RNA-seq, as a research assay, if you want. Right. That's also something we started doing now, is we are obviously trying to get patients to sign a consent to do research, to enable us to do research on the clinical samples. It's all de-identified, and so on. But that, allows us to essentially pilot things like RNA-seq in those cases. And we see, even though we can't report results, from RNA-seq because it's not a clear assay, to the physicians, right, but there's so much actionable information in RNA-seq.
So is it fair to say that that's kind of an activity that your lab undertakes then, rather than the center?
Yeah, I mean, and that goes back to the sort of, dual mission of the Institute, if you want. We have a clinical mission, but also a research mission. And so the research mission is broad. It's, discovery of novel mutations, discovery of novel potential drugs, mechanisms and so on. But it's also, trying to develop the next generation of clinical assays, trying to understand, how we should be, sort of doing things when it comes to genomic testing moving forward, and it's very clear for all of these, sort of experiments and all these, sort of, initial sort of programs that we need more than just the DNA. But that's very much again part of the mission of the Institute. I mean, it definitely sort of meshes into what my lab does, to some extent. But this is the joint mission of the Institute for sure.
Tell me more. What else does your lab work on? I know you guys have sort of an exhaustive list of tools and resources.
Yeah. So just one more thing about the Institute. I mean, in much the same line of, the DNA is not giving you the full picture. RNA is, potentially very helpful. We also have an organoid program. So that's organoids basically, more or less, cells, fresh cells coming from the patient's tumor that we grow in a dish in 3D, in a 3D format, in such a way that we can get enough cells that we can, make a collection of, sort of tumors that all come from the same patients. The 3D culture is advantageous because it sort of maintains or sort of, enables preservation of some of the features of the tumors from the patient. The tumors don't get to evolve as much if they are grown in 3D culture as if you grow them in 2D. But, the goal of this, and again, sort of proof of concept that we've established now, several times, is really to essentially create copies of a patient's tumor, but, in vitro, sort of ex vivo. That allows us to test multiple drugs and multiple drug combinations on the tumors from individual patients. And again, this is a research, concept right now, but this is something that we also see as the future of precision medicine, something that is going to be used in the future, maybe in 5 years, 10 years from now. And I think, that it has, great potential. I mean, it's just realizing that, we don't, understand everything about, the genome. It's just lots of mechanisms, lots of, reason why, cells, respond to drugs, tumors respond to drugs that we don't understand. And having this ability to carry out, almost, mini clinical trials on individual patients with hundreds of drugs, I think is really sort of, very helpful in terms of illuminating, what works and what doesn't work on patient samples and hopefully on patients down the line. I mean, obviously we see this as at some point also an opportunity to learn how to connect mutations and, drug responses, because as we see, basically, the patients respond to the same drug in the context of having the same mutation, and we can also start, connecting mutations and drug responses.
Yeah, we're running into a lot more data from spheroid experiments and actually companies that are working hard to kind of commercialize some of these models. Do you see these kinds of models as making their biggest impact close to the point of care, or can you imagine entire sort of drug discovery workflows that use spheroids maybe rather than mice? Or
Absolutely. I mean, 100%, obviously, mice have limitations when it comes to mimicking, sort of, responses of humans basically to treatments and so on. We really think that organoids more and more are going to provide essentially complementary platform for testing molecules, because essentially, in what we see, I mean, they basically really mimic in very sort of effective way, tumors from patients. I mean, what we do is, as I said, is still mostly focused on metastatic patients, and we're actually getting a lot of organoids from metastatic patients. And that's the same patient population that actually goes into clinical trials. So, I think, organoids, spheres and so on have not necessarily always, worked out in the past, but we feel it's because essentially they were often derived from, primary tumors, primary patients, and not from metastatic patients where essentially the trials are conducted in, sort of, down the line. So, what we are already seeing, quite a, bit of similarities between the way that organoids respond, to treatment and patients respond to treatment. But again, focusing on, tumors, disease that's metastatic and basically mimicking, sort of, where the clinical trials are happening. So we really think that, there's great potential, as we discussed, both as a precision medicine tool, but also as a platform for drug discovery. Because, we've actually now established organoids in about 12 different tumor types here in the Institute. And so that allows us to essentially test molecules, across those 12 different tumor types. And basically, we can tell easily, where maybe, for example, to position a molecule by essentially trying it in different tumor types and seeing where potentially it's most effective in. So we have, prostate cancer, breast cancer, colon cancer, lung cancer. We have all of the major tumor types, 12 different tumor types in total.
That's fantastic. That's really exciting.
And just one more thing that I want to add briefly is that we're also working very hard to create immune competent organoids. So that's one of the limitations of organoids, in some ways, is that, as you culture tumor cells, initially they have, infiltrated the immune cells. Those cells die out, because the medium that we use to culture cells is not ideal, for maintenance of non-tumor cells. But now we're trying very hard to essentially, get the immune cells out of the tumors, expand them, in variety of different ways, and then basically re-inject or co-culture tumor cells and immune cells from the same patients, in variety of different ways. And we're starting to get some really good results in doing so. And that likewise, I think, is going to create, models that are very, very faithful, essentially, in terms of recapitulating, tumors that you see in patients. This is really, I think, exciting area of research in our group.
Yeah, there was a paper out in mid-December in Cell from Calvin Kuo and colleagues, where they did a really nice proof of concept around organoid modeling of the tumor microenvironment.
Exactly. You're actually leveraging, sort of that information in the paper, for the research happening here. Absolutely.
No, just the amount of progress we've made is fantastic. I do think you're right in thinking there's a timeline here, though. These technologies are going to take some time to become kind of routine and commoditized, but man, how powerful.
Absolutely.
All right, so your lab, you've got sort of a dry lab component. You clearly have a wet lab component developing technologies. And you mentioned some external collaborations. So who do you reach to outside of the Cornell network to kind of help motivate this work?
Yeah, so we have quite a few collaborations with companies, pharma companies, for example, and these interactions and these partnerships actually really to me are very exciting and very informative. Obviously not only they provide a source of funding for some of the research happening here, but I think most importantly I think it's a mindset that we are essentially acquiring as a result of working with those companies, which is a translational sort of commercial mindset. Is how to take discoveries happening here at Cornell and turn them into a commercial product. How do we think about, what the industry sort of needs and wants in terms of, let's say new drugs, how do we fill the gaps that exist? I think all these interactions are very helpful in terms of understanding how industry thinks and works, and how we can, be as useful as possible in the context of these interactions. And again, how we can really, translate as many of the discoveries happening here, into commercial products, into new drugs. Because, I think that's what we, all aspire to, is really, to come up with better ways to treat patients, whether it's, better diagnostics, better, sort of mutational profiling, technologies and so on, but also how, better drugs, better ways, to keep cancer at bay.
I want to pivot the conversation a little bit and talk more about data again. So one of the recurrent themes of our conversation on this series is around advanced analytics technologies, and often we just use an umbrella category of artificial intelligence, although as that terminology gets used and misused more and more, I'd love to give you the opportunity to let's come up with a working definition for what your lab, your research group, your institute, what kinds of analytics are you applying and innovating in towards precision medicine?
Yeah, so I think that's a very important technology. We're trying to leverage it as much as we can. I have a background in computer science, and I've been doing sort of machine learning actually for a long time. Actually, I had a master's in machine learning and AI. I got several years ago. Long before AI was cool. There used to be a period of time where we would not even call AI. We actually AI would have a bad reputation. We'd actually avoid using AI, as a terminology. So I've actually lived, through those years. And, to be honest,
we might get back there yet if we're not careful.
I know, exactly. I completely agree. I think there's, we need to be very careful about all this. The amount of hype, I think, in the industry, I think is extremely high. But the reality though is that, with deep learning, especially, we're seeing really, very compelling advances in technology in terms of, using AI, building models that are very predictive, very reliable, very robust. In my view, when it comes to deep learning, I think the killer applications are very much, imaging related, basically the ability to, feed complex images, into these, sort of neural networks and basically train for, tens of thousands of images models that are very predictive. We've actually had, some of the research happening in our group, in our lab has been connected to this. We're applying AI to images here in the medical college, in a variety of different ways, whether it's, histology, slides, radiology sort of images, but also images that, maybe a bit, less sort of well used or less well known. For example, we have a fertility program here at Cornell that's, very good mode. And, in the context of a program, they've been collecting images of fertilized embryos after IVF. So we have tens of thousands of such embryos with images and detailed clinical information. And we started applying deep learning to those images, and we're getting really, very compelling results when it comes to predicting the outcomes of fertility treatments like IVF based on images of embryos after fertilization. Just an example of a project that's nothing to do with cancer, but, where we've been, essentially able to use analytics and AI, I think, in a very effective way. And we have a paper that's in resubmission about all this. In my view, I think, our utilization of AI has been most compelling when it comes to image, and the nice thing, again, is that a medical system has tons of images, generating as part of standard of care. So there's a lot of data that we can use for all these analysis. But we've, the other sort of component where AI, has been successful is in more actually in terms of drug discovery, in terms of, developing drugs, discovering drugs. We've actually, a big component of my lab is actually focused on that, basically how we can use AI as a way to speed up, improve, enhance how we develop new drugs and how we essentially characterize, new drugs. So we've had a couple of papers. One of them was, for example, using AI as a way to predict targets of molecules. For example, targets of molecules that come out of phenotypic screens. You don't know the target, but you can generate a bit of information about, those molecules. For example, treating, let's say, cell lines with, the molecule, and then seeing what cell lines die, what cell lines don't die. Transcriptomic analysis of, cell lines. You treat a cell line with a drug or with a molecule. You see what genes go up and down. This information, even though it doesn't tell you the target, if you integrate it across, let's say, the structure of, the molecule, the viability screens that you can do, transcriptomic analysis, all this information, if you can integrate it together using AI or using machine learning, really actually starts really sort of, revealing the target of molecules in a very effective way. And so we have a couple of papers about this, really sort of, nice way to use AI. And it's more machine learning and AI. It's not deep learning. It's basically, Bayesian, networks, but very effective. Really sort of, helps us narrow down to the, to the target of a molecule in a very effective way. And in my view, I think this kind of technology is going to be used more and more as, as we sort of start to understand their power, their strengths, their weaknesses. I think they can really help when it comes to specific aspects of how we discover new drugs or develop new drugs, basically filling gaps, that we have now. For example, in terms of target identification, we are also using AI. I think there's a killer application of AI in my view, which is to learn how to combine molecules and drugs. As we are getting more and more, inhibitors and more drugs, I think it's a major problem in my view to really understand how to combine them. There's just so many combinations. I think there's no way that, I keep saying there's no way that we can pipe it fast enough that we can test every possible combination. So we're going to have to be smart about it. But in my view, I think, if and we've actually shown, proof of concept of this, maybe we can test, a small number of combinations and then extrapolate, based on AI and, to the other combinations that we haven't tried experimentally. And maybe, this would allow us to, basically sort of navigate, explore the space of possible combinations in an effective way. And again, I think this is one of the applications of AI that I think has a lot of potential. So, my lab also works on these components. It's like, how to tackle difficult problems, in drug discovery using AI. And we're starting to see some nice success. But obviously there's a lot more to do.
Yeah, no, we're really interested in this problem of combinatorials as well. And it occurs to me that there's the one challenge of just the overall combinatorial space. There have been literally tens and tens of thousands of compounds that have gone into humans over the years. And that's a huge combinatorial space to just test in the lab. But more importantly, we don't have a lab system that recapitulates the human biology correctly. And so, you increase the odds of toxicity and off-target problems. And so thinking about using data integration in a smart way to kind of come up with a more, let's say, comprehensive guess at what's going to happen in vivo is probably important.
Absolutely. And again, I mean, just to add a little bit to this, we have a lot of data. We had another project to predict toxicity of compounds based on clinical trials results. We actually, people have done so many clinical trials. We know which ones, have gone all the way to FDA approval, which, failed early because of toxicities and so on. We've actually shown that we can actually learn from, these trials combining, features of, let's say, the molecules, the targets of the molecules, we can actually predict, pretty effectively, which molecules had the potential to be toxic. Again, I think this is, basically leveraging the fact that we've done so much. The field has acquired a lot of data, and we think that we need to learn from that data, and so that we don't repeat the same mistakes.
No, that makes sense. So you've talked now about obviously clinical data and patient records. We have genomics data, RNA sequencing, spheroid type information. What kinds of data do you think we're most lacking? We have got a lot of imaging data. Where would you like to see either the research or maybe even the clinical community focus their efforts on data acquisition?
Yeah, I mean, I think there's several things, right? One is that, we still live in the world of silos. And so we still live in a world where, it's hard to combine information from multiple, centers, hospitals. I think that we need to move beyond that point, I mean, because as much as, a large hospital like this one, like the one that we are in, we have access to a lot of patient samples. It's just basically not enough. It's basically, we need to combine essentially data across multiple hospitals, multiple institutions, multiple, centers across, the entire planet, essentially, so that we can get to the sample size that we can, where we can essentially make, essentially robust associations. We're not anywhere close to having that possibility. So honestly, I mean, to me, that's one of the main, sort of one of the first things that we all need to work on is how to find ways essentially to combine those data sets into, larger data sets so that we can increase sample size and increase power. I certainly do think that, we are in the situation where we've pioneered a lot of things in cancer. We can now essentially take all the things that we've done in cancer and try to apply them to, other diseases. We have programs here at Cornell focusing on Alzheimer's disease, cardiovascular medicine, diabetes, where I think there's great potential to essentially use, both genomic information, but also the same concept of organoids, right? Basically ex vivo models. Often it's going to be iPS derived, basically you get, let's say skin cells, you make iPS, and then you can, sort of differentiate those iPS cells into all kinds of different tissues. You can make a mini heart, you can make a mini brain based on iPS cells. And I think there's a potential then to try, essentially the same model that we use, for organoids in cancer. In organoids, we're trying to kill cells. Obviously, in those other diseases, we don't want to kill cells. We want to change specific phenotypes. But again, I think the same concept, I think, is very much applicable, I think, to those other diseases. And I see great potential in the future for precision medicine to essentially be applied to these other diseases. But still, basically leveraging the same concept, genomics, and then, some kind of a, ex vivo model type of, sort of program.
Let me ask you a little bit about the company that I'm only familiar of, the OneThree Biotech that's spun out of your lab. Tell us a bit more about the company, who's running it, what's its mission, and are there other companies that have come out of your lab that I'm not aware of?
Yeah, so, it's part of, again, trying to kind of sort of go to the next phase, commercialize, concepts coming out of the lab. I think it's really important. On the West Coast, it's something that's pretty natural. People come up with companies, start companies over time. On the East Coast, I think we're just getting started. There's not been the same kind of culture of starting companies that you see in the West Coast. But it's getting there. It's getting better and better. So one of the companies that we started was a company called OneThree Biotech. OneThree is interesting. It's a little short story. It's our institute is basically in Manhattan, and so we are located in 1305 York Avenue. We are actually located in the 13th floor of that building, and my office is basically office 1313. So we've actually been exposed to like 13, basically since the beginning. It's worked out reasonably well for us. So basically, as a sort of testament to all this, we've basically named the company OneThree Biotech. Basically, it's a company that's looking to commercialize, looking to use AI as a way to enable drug discovery. I mean, not a new concept in some ways, but it's leveraging essentially research happening in my lab. Basically, the research that I mentioned to you when it comes to predicting targets of molecules, when it comes to combining drugs in a smart way. We have predictive models that we think are very helpful when it comes to doing all these tasks that are very difficult experimentally. And so we're trying to now basically build a kind of a drug discovery program based on these technologies. Honestly, our goal it's not moving as fast as I would like it to move, but basically I would really like to sort of think about disrupting the whole industry, basically thinking about how we can use AI at the beginning of a drug development process or drug discovery process and not, at the end. I think there's a lot of utilization of AI that's kind of retrospective in some ways. You do, you basically have a, molecule, you have a target, you have a molecule, you basically go through clinical trials and then you have samples and then use AI as a way to maybe understand, what the molecule is doing in clinical samples. But I feel like we can potentially rethink the whole industry from the start, think about how we can use AI as a way to discover new targets, think about how we can use use AI as a way to maybe, create, new molecules. Think about how we can use AI as a way to maybe connect these molecules with, specific targets. Think about how we can use AI to predict the toxicity of molecules, in basic trials before we actually give the molecules, to humans. I think we can sort of think about rethinking the whole industry in some ways. I know it's very ambitious. It's probably not something that's going to happen anytime soon, but I'd love at least, to go through the effort, to go through the motions. So trying to rethink the whole, process essentially and see if it works, see what works, see what doesn't work.
You and I are like-minded about this, and there are quite a number of us also who are excited about the potential to make differences at various points in the life cycle of developing a new therapy. All right, one last sort of area of interest I just had to ask you. A while back, your lab developed actually a program for Oculus Rift. So this is AR, VR. This is cool. This is sort of a total departure from the other stuff we've been talking about. Tell us about that. What healthcare problems were you trying to address? Is this something you're still working on?
It's a cool, program. I have basically one fellow who's working on this. I mean, it's a small program. Again, I mean, it's one of those things I'd like it to be a bit more active, but, you have to pick your battles in life and we can't do everything. But, so it was an attempt at, essentially trying to address the topic of, information, let's say, overload. I mean, the reality is a bit like, as I was mentioning earlier, is that for every patient that we care for in this hospital, we generate a tremendous amount of information, whether it's imaging information, genomic information. We have, also access to the entire history of patients, so from the moment that they check into the hospital and so on. And, to be honest, I mean, I don't think there's a good way, or don't think we have a good way to visualize that information, and we've been maybe also trying to think about how to maybe redesign how we consume medical information in some ways. And, I really think that there's a potential for AR and VR to actually be helpful in terms of consuming medical information. It's information that's very complex. A lot of it is not relevant at a particular time point, in the care of a patient. But, again, I think ideally you would want a physician to have access to this information and be able to browse it, in multiple ways, in a very natural way, which is not essentially, the way that typical sort of electronic medical record, offers right now. Basically being able to essentially kind of use it like a medical cockpit, if you want, where you have access to the entire set of information about a patient and browse it. A bit like Minority Report, the movie about with Tom Cruise, looking at different pictures and so on, basically manipulating this information with his hands. And you would like to be able to do the same thing with medical information. And I really think that there is a role to play for VR and AR in this. We basically established a little proof of concept, on this. We've put a lot of medical data in those, sort of virtual and augmented reality sort of, systems, radiology data, but also, genomic data, network data showing, sort of connections between, mutations that you see in the patients and potential drugs that you can use for the patient. And we're just testing different things. We're trying to see what works and what doesn't. It's early stage, and the systems are getting better and better. I'm always, still blown away by, the HoloLens, which is one of the main systems that we use now. It's basically augmented reality. You see virtual objects, that sort of almost interact with the real world. It's very cool stuff. And it's early days. I think that, next 5 years, 10 years, we'll see much better, sort of systems, very like almost like glasses that you just put on and that you see all these virtual objects. And we'll have, I think, to reinvent how we interact with, data in this way. I mean, we'll have the possibility to reinvent, how we interact with data. I keep saying, the way that we consume data now is basically mostly, two-dimensional, which is basically on a screen, which is essentially not, a strong deviation from the book, more or less. And I keep saying the book is probably not a strong deviation from like a wall, like in a cave. I think that we can also rethink how we, manipulate, consume, data. And I think those systems, the HoloLens and, the Oculus and so on are going to be great opportunities for rethinking how we consume medical data. I'm hoping to play a role in this and but we'll see how that goes.
That's absolutely fascinating to me. I wish you luck with that as well.
Thank you.
Olivier, this was a ton of fun. I could keep you all afternoon, but I know that we're all busy people, so I'll let you go. Thank you for spending some time with us. To our listeners out there, this is Olivier Elemento, who's really one of the real practitioners in the space. I'd love to stop by next time I'm in New York and see what you guys are working on.
We'd love to have you, and thank you so much, for doing this. It's, it's been a lot of fun. So
great. Thanks very much.
This has been episode 7 of Talking Precision Medicine. Please share it with your colleagues, leave a comment or review, and stay tuned for the next one. Thanks for joining the conversation.